Du coup, mon organe d'intérêt, c'est le muscle. Et donc, pour ajouter du challenge au challenge, j'avais prévu de faire la présentation en anglais en espérant que ce soit OK pour vous tous. Et c'est bon, ça fonctionne. I like to begin with, I like to say that skeletal muscle is to me the most important organ in the body. First of all, because it accounts for up to 1/3 of the body weight, and also that the truth is that you have more than 600 muscles in your body, whereas you only have one brain, one lung, and so on. So I think it makes it for this important organ. So of course, it's an organ which is indispensable to locomotion, blinking, so muscular physiological functions, but it also has extramuscular functions such as heat production or glucose homeostasis. Je vais me prendre un petit pointeur, sorry. And the organization of this tissue is really fascinating. Actually, it looks like a babushka doll. At the level of the organ, the muscle is surrounded by a fibrous membrane, which is called epimysium. And this organ shelters actually several muscle fascicles or also muscle bundles, which are surrounded by the perimysium. And these bundles shelter several muscle fibers, which are surrounded by the endomysium, which is the extracellular matrix of the muscle tissue. And each individual muscle fiber is surrounded by the sarcolemma, so the plasmic membrane. And each fiber shelters several myofibrils, which themselves are composed of a succession of several sarcomeres. And the sarcomere is truly the functional unit of the muscle cell, the contractile unit. You can see one sarcomere here between these two Z lines, and it's responsible for the cell contraction with the sliding of the actin filament here towards the myosin filament here. And what's important to note in this organ is that in vivo, it's attached to the bones through the tendons. And this anchorage point allowed by the tendons is really pivotal for muscle development and growth. And it's also something which is very important in 3D models. We will talk about this later. So if we go back to the cellular level, besides the myocytes, the myofibers that we just talked about, you have many important cells in this tissue. Of course, you have vascular cells, which are responsible for nutrient. They bring the nutrients to the tissue. So you have endothelial cells, pericytes here. You also have immune cells, which are really pivotal for muscle regeneration. You have pro-inflammatory cells for the inflammatory phase, and you have regenerative cells for the resolutive phase. You have interstitial cells, such as telocytes. But actually, the most important cell of them all is hidden here, right beneath the basal lamina of myofibers. So it's really stuck to the myofiber, and they are called satellite cells. They have not been discovered that long ago, only in 1961, and they are really the skeletal muscle stem cells. They are responsible for the muscle regeneration. And they become activated in case of disruption of basal lamina integrity. So that's why they are really hidden beneath this basal lamina. And once they're activated, they proliferate at the myoblast stage. Then they elongate as myocytes. Then they fuse together. They become a myotube. And then this myotube will either fuse to the damaged myofiber, such as the one that you see here, or will become a fully grown myofiber, which will replace the tissue. So these are really the center cells in the muscle tissue. So all of this organization and in the cell-cell interaction state is what happens in physiology. But when things go wrong, what can you get? You can get many diseases from the one single organ, and you can categorize those diseases into three main categories. So you have the genetic diseases, the acquired one, and then you have the gray area of diseases which are probably partly genetic and partly acquired, but they still have a very various feature in the muscle. So a quick word about the genetic disorders, because they maybe are the most well-known ones. Probably you know some of them with the Téléthon advertisements. And the most well-known is the Duchenne's muscular dystrophy, which where you have excessive fibrosis in the muscle tissue. The acquired disorders are mainly traumatic or toxic, so I won't go further into detail for this one. And the partly genetic, partly acquired disorders are actually a vast category of diseases. You can have neuromuscular disorders such as amyotrophic lateral sclerosis, which is actually not a muscular disorder itself, but a neurological one, and then the muscle tissue suffers from the lack of innervation. You have autoimmune disorders, and you have the wide range of skeletal muscle wasting disorder, that is to say a loss of muscle mass and shape, a loss of muscle tissue, which is either associated with aging, and then it's called sarcopenia, loss of muscle mass and force, or associated with cancer, and then this active loss of tissue is called cachexia, or with chronic inflammatory diseases such as chronic obstructive pulmonary disease. And this last topic, COPD associated sarcopenia, is actually my topic of interest. So just a quick word about chronic obstructive pulmonary disease, COPD, because it's a disease that possibly not all of you are familiar with. It's a primarily lung disease, which is usually associated to inhaled particle exposure, such as cigarette smoke, as you can see here. It's also known as chronic bronchitis. And actually what the aggression of cigarette smoke does is that you have a dual lesions in the lung. You have the bronchial obstruction, which is caused by excessive mucus secretion and peribronchial fibrosis. And you also have an alveolar destruction in your lung, which is also known as emphysema. So I said it was a primary lung disease, but actually the features of this disease extend far beyond the lung. And in some patients, you also get muscle damage, so COPD-associated sarcopenia, which corresponds to 4 cardinal features in your muscles. First, you have a muscle atrophy at the level of the organ, but also at the individual myofiber level, so smaller myofibers, smaller muscles. You have also a decreased vascularization of your muscle. You have an increased fatty tissue infiltrate, and then you have also a loss of your number of mitochondria, which is associated with a loss of oxidative myofibers and overall a decreased oxidative phosphorylation capacity. So, okay, you can get muscle damage in addition to lung damage, but is it bad? Is it serious? Well, actually, it is pretty bad within a disease which is already pretty bad itself, which is responsible for a lot of death through the world. Wow. And actually, when you do have sarcopenia and you are a patient with COPD, you really have a higher mortality risk than if you do not have sarcopenia. For example, if you see this survival curve here of patients here with COPD but without sarcopenia, and here with COPD and with sarcopenia, you see a difference in the mortality risk up to 10 or 15 folds. So the question is, how do we prevent this patient from going from here to here? How do we prevent sarcopenia from happening? So for this, we need to understand the initial pathophysiological mechanism, and for this, we need relevant models. And of course, it's not in this webinar organized by the GDR Organoids that I need to convince you that mice models are maybe not the most fit models to study this question. And of course, we would like to have a relevant 3D model built with human cells, because you are all well aware that mice are not mini humans. And the research question I would have for my specific project would be how to model properly COPD-associated sarcopenia, and which cardinal features should I choose among those, if not all. So that leads to the next question, which is, which 3D model should I choose? Which shape, which functionality, which cell representation? And that does lead us to the fascinating field of strategies of 3D skeletal muscle modeling. So you see, I chose to represent them as a bingo because as you can see for each of those parameters here on the left, cell origin, cell complexity, 3D model strategy, or mechanical strain, you have several possibilities that can be all combined. So you have quite an infinite number of different models that you can get. And it's a very simplistic representation, so I apologize for this. For example, if you look at cell origin, you have a lot of models which have been built with a very famous cell line of murine myoblasts, which is called C2C12. You have some models which are built with primary satellite cells, either from healthy tissue, either from diseased tissue, even if it's harder to grow muscle cells from diseased tissue. And you have a lot of models which have chose to grow cells from iPSCs because it would then you have a better proliferation than disease satellite cells, and they drive the iPSCs toward the myogenic lineage. If you have a look at cell complexity, basically the models either try to include the vascular component, and then they would include endothelial cells, sometimes pericytes. Either they want to go towards the neurological component, and they try to model the neuromuscular junction by adding motor neurons. And some models actually do both. If you look at the 3D model strategy, so of course this is oversimplified, but I categorized it into 4 different categories, which are the tissue engineering field with bio-artificial muscles construction, the microfluidics field, the bioprinting and micro patterns field, which are not the same, but let's say in the idea of controlling the location of your cells, and of course, maybe the most important of those organoids, wink, wink. So, but actually the most important component for 3D skeletal muscle modeling would be the mechanical strain addition, because if you remember, in vivo muscles are attached, physically attached to very hard structures, which are bones, so the tendons, and it seems to be critical to reproduce this kind of mechanical strain if you want to properly model the organ. So I identified three possibilities. Either you can provide an anchorage point to your cells, such as to model the bones. Either you can provoke contraction in your cells, and you can do this electrically or biologically or with optogenetic light stimulation. Or you can mimic eccentric contraction, in other words, stretching, by longitudinally stretching your cells, and you can do this in a static fashion or in a cyclic fashion. And so, as I said, maybe this strategy is the most important for the muscle tissue in particular, because if you lose mechanical strain, and that happens in real life when you lose some gravity, the muscle tissue is one of the first to really suffer and lose rapidly mass and shape after a loss of mechanical strain, even if it's not very obvious in this picture here. So it seems that to conclude that the major challenge is in muscle 3D models is to obtain a terminally differentiated model, and terminal differentiation is achieved when you identify a sarcomere in your cell, which shows that your cells are able to function like in vivo. And so obtaining a sarcomere in 3D model, it's like a golden ticket from a Willy Wonka's story. It's really the major achievement I think you can have. And the key to this achievement seems to rely within the proper mechanical strain addition. So next, I want to present 3 different models. So as I said, there are many different models in the literature. There are many very, very nice papers. I just chose to represent 3 very different models so you can see what you can get. And the first one will be the organoid model. And there was this very recent paper published in eLife, I think last month, showing the last version of muscle organoid model development. And to achieve this, the authors embedded undifferentiated iPSCs within matrigel to get some spheroids, and with chemical cues, they achieved to recapitulate the different steps of muscle embryonic development. From the neuromesoderm to the neural tube induction, and then at the end they get the dermomyotome, so the future myogenic tissue, and the future neural plate. So this kind of model is more intended to model maybe the neuromuscular junction development. And when they looked at the protein level and did some nice stainings, you can see that they achieved to have myogenic cells here, which are stained in red with a myosin heavy chain. They also have skeletal muscle stem cells, satellite cells, with the PAX7 staining. And from the neurological point of view, they did identify some neural crest progenitors with the PAX3 and SOX10 staining here. So they have both myogenic progenitors and neural crest progenitors. They did a characterization at the multicellular level, and they showed that they have the nice representation of several different skeletal muscle cellular subtypes, so with single nuclei RNA-seq. So they had all stages of myogenic cells development, from the progenitor stage to the myoblasts, myocytes, myotubes. And they also had neural progenitors and another type of progenitor, which is very interesting, which is the fibroadipogenic one. And they even had some kind of function in their organoid, because when they stimulated it with acetylcholine, which is a nicotinic agonist, they did record some nice calcium influx, which is supposed to precede contraction. So showing that they achieved to have some kind of differentiation. And cherry on the cake, they actually achieved their golden ticket because they evidenced the presence of sarcomere, both by a second harmonic generation imaging, and also here by a transmission electron microscopy, and you can see the Z lines here. So this was a very nice model, but it did not reach the late stage of differentiation, probably for all cells. Probably, I think that only a few cells show the sarcomere. So as the authors underlined, it's more designed to study the different steps of muscle embryonic development rather than to mimic a fully grown organ. So this model, to recapitulate, used iPSCs modeled rather than neuromuscular junction and was an organoid. And now I would like to show you a second model, which was also originated from iPSCs, but driven towards the vascular and neurological component and using the tissue engineering field, because it's a technique with the anchorage point, which has been vastly published and it looks really interesting for the field of 3D muscle modeling. So this was also a very nice paper published a little earlier than the previous one. And from the iPSCs, they separately drove the 4 different lineages they wanted to include in their model. So they drove towards the myogenic lineage, towards the endothelial cells, pericytes, for the vascular component, and also towards neural progenitors. And then they embedded all of those 4 lineages within a hydrogel made notably of fibrin, and they let this hydrogel polymerize in a sort of hole where you had 2 little pillars here that you can see more closely here. And those pillars nicely mimic probably the anchorage point in vivo, so the bones, and provide the mechanical strain that seems to be sufficient for muscle differentiation. So if we also look at the protein level in this model, and we take a look at the vascular component, so they have first of all, they have a nice myosin heavy chain staining with a nice elongated shape, showing that probably they have differentiated myotubes. We see multinucleated myotube like we should see. You see endothelial cells in purple, you see pericytes in green. And for the neural component, you still see your muscle cells in gray this time, and some motor neurons that you can see here in red. And they even had some staining for the neuromuscular junction with the bungarotoxin staining here. And looking at the titin staining here, which is a sarcomeric protein, you can guess some Z striated shape of the tissue, which indicates a possible presence of sarcomere. And sure enough, they also had their golden ticket with the identification of sarcomere in this paper with electron microscopy as well. So also a nice paper. And as a third and last example, I would like to show a model made of with a microfluidics technique this time, and using primary cells, which is a little bit more rare in the field. And so this was also a nice paper published the same year as the previous one. And the microfluidic setup here used little hole in which there were seeding cells in order to obtain one individual myofiber per hole, if I understood well. And so they seeded within this hole, these pores, they seeded muscle cells, they seeded some fibroblasts in order to get some matrix, to get some cohesive fibers. And they also seeded vascular cells, endothelial cells, and pericytes. And again, when you look at the protein level, you see a nice level of differentiation with the myosin heavy chain staining here. And you also see that the vascular component is well represented with endothelial cells in green here. And you see that the endothelial cells form a network which seems to connect with the muscle fibers, showing that they somehow achieved to have a nice, maybe vasculated, but at least a nice model with a nice cellular complexity. And there's in this paper, the authors used this model to model Duchenne's muscular dystrophy. And what they did is that they introduced fibroblasts from a Duchenne's patient, which was is a nice idea because it's patient which have a dystrophic muscle tissue. So you do not want to take muscle cells directly from this patient, but taking fibroblasts is a nice idea. And they showed that by introducing fibroblasts from diseased patients and measuring the concentration of several ECM components, you had excessive production of ECM proteins such as collagen and fibronectin, which is a nice readout for a dystrophic muscle. I just have to say that in this paper, I saw no sign of sarcopenia, so nobody's perfect. So that leads, so all these three examples are really nice, and that's that leads us back to my research project, where actually I was not sure I could use any of these. The model I wanted to use actually was not to model muscular dystrophy, as most models are doing, but muscular atrophy, which is a little bit different. And so I was looking for a model which in which I would be allowed to control precisely the diameter of the muscle bundle so that I could evidence an atrophy later on. So which 3D model can I choose for this? And we have the chance here in Bordeaux to collaborate, and this collaboration has been initiated by the head of our group, Isabelle Dupin, with a very nice biophysics laboratory, very famous, led by Pierre Nassois, who invented a very nice technique to encapsulate cells. And this technique actually takes inspiration in the flavor pearls of Thierry Marx, who is not a researcher, but a French cook, who invented this flavor pearls by encapsulating flavors within a solid shell of alginate. And so the idea of the system developed by our colleagues is to encapsulate not flavors, but cells within a microfluidics setups where you co-inject a cell suspension, a buffer solution, and alginate on the outside, and you make some drops with cells and alginate, and those drops fall into a calcium bath. And as soon as the alginate is in contact with the calcium, it instantly reticulates, so you get nice capsules with cells inside. And just by tuning, I'm not sure it's going to work. Tuk, 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 tuk. Flèche. Tic, tic, tic. Ça marche pas. Tristesse. Je voulais vous montrer une vidéo, mais j'ai bien l'impression que ça ne va pas marcher. But okay, let's it's okay. Just by tuning the speed of the flow, and you have to imagine here that drops are falling quite silently, quite slowly, and here a little bit more rapidly, and here the drops are falling so rapidly that when they are touching the calcium bath, they are actually forming tubules and not capsules. And this tubular shape is actually quite relevant for a muscle model. So, ben voilà, il suffisait juste de faire ça. So the idea of the Samurai project is to develop a muscle 3D model which would be suitable to study COPD-associated sarcopenia. And for this, I have the chance to be able to collaborate and work with Gaëlle Rocher. She's a researcher in the lab I was talking about, and she's also she also knows very well the skeletal muscle tissue. So the idea would be to use the microfluidic system to encapsulate human primary satellite cells and to observe the differentiation, progressive differentiation over time of those cells within the alginate hollow core. And the the idea would be that we hope that the anchorage point that would be provided by the alginate scaffold would somehow mimic the perimysium, the envelope that surrounds the bundle of myofibers, and maybe provide the right mechanical cues for the cells to differentiate. So as you can see, one experiment can give a very, very long tube, and you can have meter-long tubes from one encapsulation. So it's really nice. It's really a powerful system because then you can cut the tube in several pieces, and you can perform RT-qPCR, immunostaining, Western blotting on a single experiment. So it's a really nice system. And I'm showing just some preliminary results that we obtained thanks of the help of Elise, our engineer, and Mathilde and Leila, our M1 students this year. And so we encapsulated murine C2C12, so the murine cell line, into the microfluidic system. And we wanted to test 3 different encapsulation conditions. One where we injected the cells without any matrix, just the cell medium. One where we injected the cells together with collagen in the system, and one where we injected the cells together with matrigel. And what you're looking at here is one magnification of one part of the tube. You have the alginate outer wall here. The inner wall is here, and so the alginate thickness is this size, and inside you have the cells. And strikingly, you had a very big difference between the 3 conditions. You can see that when you do not put any matrix within the cell suspension, the cell immediately organize in form of smaller balls, which is actually not what you want to achieve when you want to model skeletal muscle, and is possibly due to excessive cell contraction. And so they are not organized within a bundle, and they also do not adhere to the inner tube walls. So that was not the idea. When you use collagen, the cells nicely organize within a bundle, but they also do not adhere to the inner wall of the tube, and probably this is also due to excessive contraction. But instead, when you use matrigel, you have a nice adhesion of the cells to the inner wall of the tube, and this could be a nice signal for regular bundle of cells. So we looked at some differentiation marker following this preliminary experiment, and I'm just showing here one myogenic regulatory factor, which is required for satellite cell differentiation, and troponin T is one component of the sarcomere. And you see that along time, in the 3 conditions, we had a nice increase of expression in these 2 markers, but we had some differences in the amount of expression between our 3 conditions, and quite surprisingly, the condition without matrix was the one that gave the most expression of the differentiation marker. But these data are very, very preliminary, and they will need to be completed. But still, we had an increase a long time, which was quite promising. And then we also wanted to look at the protein level, and Elise did some very nice immunostaining and 3D imaging. We first looked at the myosin heavy chain expression, and we were very happy to see a nice elongated expression of myosin heavy chain in our myotubes, showing that maybe we had differentiated myotubes inside the model. And we also wanted our Gödel ticket, so we looked at alpha-actinin expression, and although we can see that we do have some alpha-actinin, so it's supposed to mark the Z lines of the sarcomere, we do have some expression within the cells, but sadly there is no Z line observed here, so no golden ticket for us yet. But of course, the project is at the beginning, so the next question would be, how can we increase the level of differentiation of our myofibers within this model? And as we discussed, we can use several strategies for this, and we could use, for example, electrical stimulation, or we could just simply use static stretch in a system similar to the one that I presented, where we could glue the tube to 2 little pillars. This has to be discussed yet. And not to forget that actually my question was how can we model COPD-associated sarcopenia? So I also have several strategies that could be used here. I could first use corticosteroids exposure, because corticosteroids are a very well-known inducer of muscle atrophy. They directly bind to the glucocorticoid receptor in muscle cells, and they directly activate protein degradation through the UPS system and autophagy. So it's a strategy that could work. And I could also use hypoxia, it's another well-known inducer of myotubes atrophy. And you can see here as an illustration that when you expose C2, C12 myotubes to severe hypoxia, that is to say 1% of O2 for 3 days, you get a dramatic decrease in the differentiation because you have a dramatically reduced numbers of myotubes, but you also have a thinning of the remaining myotubes with a reduced diameter, so myotubes atrophy. And actually both strategies, corticosteroids and hypoxia, are relevant for COPD. So still ongoing work. And so humble conclusion from what I've gathered from this field in which we are at the beginning is that you dispose of many different models that use very various techniques. And so it seems that there is no ideal model for skeletal muscle itself, and that the choice will depend on the specific biological questions you have, which really depends on the cardinal features of the disease you're trying to model, and of course the partners you can work with, because it's always nice to work with local forces. So we are really lucky to work with the team of Pierre Nassois and Gaël Rocher at the BioFlav and the Voxel platform. And I'm also really lucky to work in a group who gave this group a great impulse for 3D models, led by Professor Dupin. I was really lucky to have the help of Elise and also Leila and Mathilde for the beginning of this project, and all of these colleagues, which I love very much. So thank you to all my colleagues for their help, and thank you to you for your attention.
So thank you very much. It was really nice. Do anyone have any questions?
That can be in French
now?
also.
In French also, if you want to.
Je vais bien poser une question, si c'est possible.
Oui, bien sûr. Et on peut, en français, c'est aussi bien, je pense.
Ouais, c'était vraiment magnifique, votre exposé.
Merci beaucoup.
En fait, je trouve quelque part dommage que vous ayez passé beaucoup de temps et que vous essayez de faire des choses hyper intéressantes avec un modèle C2, C12, alors que vous avez à l'hôpital la possibilité d'avoir des cultures primaires, des cellules satellites de patients humains, ou qu'il y a des cellules satellites de souris qui sont disponibles. Ça ne demandera pas plus de travail de mise au point et les résultats seront 1000 fois plus jolis à l'arrivée. Encore plus joli, excusez-moi, que ce que vous avez avec les cellules C2, C12. Donc, en fait, c'est pas tellement une question que j'ai à vous poser, mais un conseil. En fait, investissez du temps dans les cultures primaires plutôt qu'avec ces cellules C2, C12, parce que de toute façon, ça vous prendra du temps, et à l'arrivée, le temps que vous aurez passé vous donnera des résultats beaucoup plus pertinents, je pense, avec toutes vos techniques. En tout cas, ouais, c'était vraiment superbe, bravo.
Merci beaucoup. Et enfin, merci beaucoup déjà, j'apprécie beaucoup. Et puis, bien sûr, merci beaucoup pour votre remarque. Vous avez évidemment tout à fait raison, parce que toutes ces mises au point, en fait, après, elles sont plus tellement relevantes quand on passe sur des cellules primaires, et en plus qui n'ont pas la même taille, parce que les critères de taille pour les mises au point sont importants. C'est vrai que c'est quelque chose, évidemment, qu'on voudrait faire, mais ça fait que très récemment que j'ai accès à du tissu musculaire humain avec le CHU. Et en fait, j'essaie justement de mettre en place une convention avec le service d'orthopédie pour avoir accès à du tissu frais, faire pousser des cellules. Et effectivement, c'est quelque chose vers lequel il faut qu'on se tourne au plus vite, oui, bien sûr.
Merci.
Merci. Et je crois qu'il y a une main levée, mais peut-être, je ne sais pas si c'est un bug ou pas, Donata Yandolo.
Oui.
Bonjour.
Oui, bonjour. Désolée, je n'ai pas de caméra sur cet ordinateur. Merci pour cette présentation. Peut-être I switch in English, so I'm going to be more...
It's fine.
I really enjoyed your presentation, very nice. I had a question about the technique that you were presenting, like to create the tubular structures. I was wondering whether there is a way to keep the tension, because I can imagine that whenever you cut, you make the pieces out of the longer tube, you're gonna have a relaxation, let's say, of the material within, and therefore the mechanical, the mechanical cues might be lost in a way or reduced. So I was wondering whether you had any, any comment on that or any strategy, let's say, to keep that.
Yeah, of course, you are perfectly right. If we will change the cues if we cut the tubes. So it's something that I have not started yet, but I would imagine two different strategies. Either I keep the tube in culture and perform the electric stimulation that some labs do with just electric impulses over the whole culture. So hopefully it would transmit to the whole tube, even if it's quite long. Either it has to be determined before that the tube is cut into same size pieces and the same tension is applied to all the pieces, and then it's kept until the end for this specific experiment. For example, for the static stretch strategy. If you have other suggestions, of course, I'd be happy to hear them because it's the very beginning of the project. So.
Okay, thank you. I'm gonna think about it, but I thought, that it was something, let's say, important to
of course
keep in mind. And I've been working mostly with electrospinning techniques, and there, I mean, like there was a way to creating like like something like a frame, and so the stretch, so the fibers, the polymeric fibers would have been stretched either by the spinning disc or by the frame, and therefore the length would have been imparted by the device that you were using. Here, I don't know. I might give it a thought, but I do not have any.
No, it's really interesting. I'm not expert in microfluidics at all, really. So I'm not sure how the technique, the encapsulation technique, is compatible with electrospinning, which is something which I believe it's a little is different. But I have to admit that I'm really ignorant in this matter. So I will speak with Gaëlle and Laetitia, the head of the platform, about this.
Okay. Congrats anyway.
Thank you very much.
On peut faire encore une dernière question si vous voulez. Équipe 2, ils ont levé la main tout à l'heure.
Oui, mais merci. Mais c'est en fait, elle vient de répondre, c'était est-ce qu'il y avait une stratégie pour exercer une pression sur les fibres là qui ont été joliment synthétisées. Mais merci beaucoup, c'est une excellente présentation. Merci.
Merci beaucoup, vraiment.
Très bien. Dernière question à Monsieur Dupont, du coup. Allez-y.
Oui, bonjour. Merci pour votre présentation déjà, c'est très prometteur.
Merci.
J'avais une question, j'ai peut-être mal saisi votre process. À un moment, vous faites intervenir du calcium pour la gélification. Or, le rôle du calcium dans le muscle est
important.
Est-ce que vous n'avez pas une inquiétude quant au fait que vous allez peut-être biaiser le système ou encapsuler vraiment trop de calcium à l'intérieur de la construction, ce qui pourrait provoquer des contractions spontanées
C'est vrai que moi, je ne l'avais pas effectivement envisagé comme ça, mais en réalité, comme effectivement, il y a, dans certains settings, il y a une contraction très importante des cellules alors qu'elles ne sont pas encore différenciées en myotubes. Probablement, le calcium joue un rôle, mais effectivement, c'est le calcium qui fait réticuler l'alginate. Donc, si on ne met pas de calcium, on ne peut pas avoir cette enveloppe. Et par exemple, plus tard dans la culture, si on enlève le calcium, l'alginate se dissout, ce qui peut être pratique pour d'autres applications. Mais effectivement, ça pourrait contrôler le niveau de calcium, serait quelque chose qui pourrait être intéressant pour le système, bien sûr. Et après, ça n'empêche quand même pas d'observer un éventuel influx de calcium dans le système. Mais je sais que vous travaillez, enfin, j'avais vu votre nom, je sais que vous travaillez aussi là-dessus, sur ces modèles. Je pense que vous êtes plus avancé que moi, donc j'ai hâte de voir ce que Clémence fait aussi.
C'est une approche différente, mais c'est vrai qu'on est en train de travailler sur le rôle du calcium dans le milieu pour la maturation, et a priori, ça a un rôle assez important quand même.
Et c'est des concentrations, parce que du coup, c'est des concentrations qui sont entre guillemets extra-physiologiques quand vous travaillez dessus, plus que ce qu'on met dans les milieux de culture, quoi.
Ouais.
D'accord. Merci en tout cas.
Parfait. S'il n'y a pas d'autres questions, on va avancer avec Clémence Lièvre, du coup, qui a fait des études de pharmacie et qui est maintenant doctorante à Nantes, je pense dans l'équipe de Monsieur Dupont, du coup. et qui va vous parler des organoïdes des muscles squelettiques aussi, mais avec le version de thérapie génique avec des AAV du coup.
Alors déjà, est-ce que vous m'entendez bien?
Très bien.
Ok,
Merci.
parfait. Est-ce que vous voyez mon diapo correctement?
En mode présentation.
Ah. Toujours en mode présentation?
Non, là c'est bon. Très
Là,
bien.
c'est bon? Ok, parfait.
Allez-y.
Et ça passe les diapos. Ok. Eh bien, merci pour cette présentation, cette introduction, et merci de me permettre de présenter mon projet. Donc, je vais aussi faire ma présentation en anglais aujourd'hui. Et je vais mettre ma caméra. So I'm Clémence Lièvre, a PhD student in the Next Generation Disease Models team in Target Lab in Nantes. And today I will present to you the foundation stones of a preclinical testing platform based on skeletal muscle organoids that we are creating in the team and that will help us to develop safer and more efficient gene therapy strategies for neuromuscular disorders. The team is interested in Duchenne muscular dystrophy, DMD, an X-linked genetic disease caused by a mutation in DMD gene coding for dystrophin, cytoskeletal protein essential for maintaining the structure of muscle cells. Its deficiency causes muscle degeneration in young boys, which progresses into respiratory failure, cardiomyopathy, and premature death. And to date, the treatment is essentially based on symptomatic therapies. Only 2 treatments, Elevidys and Translarna, have received a market authorization to treat the root cause of the disease, but they concern only few patients with specific mutations, and they are very expensive. So there is a real need for widespread curative treatments for DMD. And among the promising approaches currently investigated in the clinic, our lab is interested in gene therapy using vectors derived from the adeno-associated virus, the AAV. The therapeutic transgene can be the therapeutic transgene package in this AAV viral capsid can be carried in the target tissue and expressed in the long term. And in the context of DMD, the proof of concept was obtained during preclinical studies in animal models, such as the MDX mice, DMD rat, or GRMD dogs, with an almost complete phenotypic rescue maintained for several years following the AAV injection and a good safety profile. However, when these products were translated in clinical trials, despite promising intermediate results, some patients have developed serious adverse events that were not anticipated in animal models, suggesting that interspecies differences remain and hinder the translation to human patients. And in addition, high doses of vectors are required, and this increases both the risk of toxicity and the cost of treatments. So we believe that in addition to in vivo experimentation, we need new preclinical models directly derived from human cells to rapidly screen more efficient AAV vectors and to develop new therapeutic strategies. So for this purpose, our strategy in the team is to use in vitro model using human-induced pluripotent stem cells. As you know, these iPS cells have several interesting properties. They are pluripotent, so they are able to differentiate into any cell type, making it possible to study the multisystemic aspects of pathologies. They are able to self-renew, and most importantly, they are directly derived from the patients. So in the team, we use these iPS cells to make skeletal muscle cells in a dish. We have cell lines derived from DMD patients and from healthy donors, and we use a published protocol to differentiate them into myotubes by recapitulating the key developmental steps from pluripotency to somite and myogenic progenitors. These cells can be isolated after 30 days of differentiation and used to then generate striated and multinucleated myotubes, which express typical muscle markers such as alpha-actinin, as you can see in this image. And we can also expand these myogenic progenitors and make large cell banks to then generate human skeletal muscle organoids in a 3D motion in an attempt to better recapitulate the structure and the function of the native tissue. And that's what I'm going to show you next. So to develop these skeletal muscle organoids, we use a setup called Montaré and commercialized by Curie Bio. So iPSC-derived myogenic progenitors are embedded in a hydrogel composed of matrigel and fibrin, which is then molded between two posts, one rigid and one flexible. And this two-post system comes placed in a 24 well-placed format, enabling the production of dozens of organoids in parallel. Thus, we can obtain skeletal muscle organoids that we can use for several applications, such as developmental biology and myogenesis studies, phenotypic characterization for neuromuscular disorders, and a preclinical testing platform for gene therapy. And this is what our project is about. So first, we designed a reporter AAV vector to prove the feasibility of a gene transfer in our skeletal muscle organoid model. We selectioned the artificial SPC512 promoter already used in the clinic to be activated specifically in muscle cells. And as a transgene, we chose the murin-secreted embryonic alkaline phosphatase MCU as a reporter. It's a modified enzyme that is released from the cells in the culture media, and its catalytic activity can be followed by luminometry, so we can assess the transgene expression for as long as the organoids are maintained in culture. And for the capsid serotype, we first chose the AAV serotype 6 because it's known to be effective in muscle cells and to be quite efficient in vitro. So after validating this construct on 2D myogenic progenitors, we used it to determine the optimal condition to transduce our skeletal muscle organoids with an AAV. So for this, we use an optimized experimental design to test several parameters in parallel, such as the cell status at the time of transduction, which means are they in proliferation or in differentiation mode, and the vector administration procedure, which means do we add the AAV in the media or encapsulate it in the hydrogel. So we tested several combinations of these parameters, and as you can see on this kinetics graph, some conditions are more efficient than others. So just to make a long story short, we performed a Pareto analysis to determine the optimal transduction conditions. And here we can conclude that the transduction efficiency is higher when AAV was included in the hydrogel and when cells were in the differentiation media. We also mentioned the fact that the dose of AAV vector is a crucial issue to work on. In fact, we know that therapeutic benefits are obtained with no less 10 to the 13 or 10 to the 14 vector genome per kilogram. And in vitro, we always need 10 to the 5, 10 to the 6 vector genome per cell to detect transgene expression. So here, we determine the minimal effective dose required to detect MCU expression in our 3D models. And here, in this first graph, we can see that there is obviously a dose-response trend, but also that transduction with 10 to 3 vector genome per cell, a transduction at 10 to 3 vector genome per cell of AAV, still allowed to detect transgene expression above the threshold, which is best observed in the second graph with data obtained at the endpoint of the experiment. So it seems that our 3D organoid model requires less vectors for in vitro experimentation. So after we analyze the ability of this model to detect long-term transgene expression, and here we can see that the transgene expression is detected up to 1 month after AAV transduction, even with low doses of AAV vectors, such as 10 to the 3 vector genome per cell. It might be longer, but we have not tried to keep our organoids more longer than 1 month so far. More than that, it seems that the transgene expression is stable during this time frame, even if it seems more complex with highest dose at 10 to 5 vector genome per cell. And lastly, we compared the transduction efficiency of 8 natural serotypes at 10 to the 3 vector genome per cells, and we analyzed the transgene expression during 10 days after AAV transduction. Here we can see that serotype 8 and 9, currently used in the clinic for muscle gene therapy, are effective, but that serotype 1, 2, and 6, for example, are 10 or 100 times more efficient than serotype 8 and 9, and these serotypes are already known to be effective in vitro. So it seems that we stay in an in vitro context with our organoid models. However, these are only preliminary results, and which will be reassessed when our organoid models are more mature. So finally, we can say that our skeletal muscle organoid model is well suited for gene therapy preclinical testing for several reasons. They could be used for less expensive and faster tests with low doses of AAV vectors. Of course, we are not going to replace entirely the in vivo experimentation, but for specific application, organoids could provide an alternative. And this is in agreement with the 3R principle for the reduction and the replacement of animal model experimentations. In addition, the organoids we have made so far are a good representation of the in vivo context of human muscle, even if we always try to make it better and more physiologically relevant. And in addition, we hope that will help us to perform high-throughput screening for new therapeutic molecules and vectors. But before we use it for drug discovery, we still have some work ahead of us to improve and validate our preclinical testing platform. Here is our few projects that our team is currently working on. So the improvement of organoid maturation level with physicochemical cues, such as electrical or mechanical stimulation, the characterization of the DMD phenotype in organoids. Then, of course, we will try to correct this DMD phenotype with an AAV vector expressing therapeutic transgenes, such as microdystrophin, for example, for the DMD. And altogether shall prove the relevance and the utility of a gene therapy preclinical testing platform to develop new therapeutic strategies and to screen the most promising AAV vector candidates. So I would like to thank all the people involved in the project, the Target Lab, our collaborator in Seattle, our sponsor, and I thank you all for your attention. Don't hesitate if you have any questions.
So thank you very much for the nice talk. We will see if there's any questions.
Perhaps,
Ça peut être en français aussi.
oui, en français. Okay. Can you hear me?
Yes.
Yes. In fact, I'm not specialist at all of AAV, but I thought that there are new AAV, neo AAV, so which are more efficient to transduce, muscle cells, and I wonder whether you will test them in your organoids.
I don't know, so sorry.
En fait, il y a des nouveaux AAV donc qui ont un tropisme
musculaire,
donc qui ont été isolés, caractérisés récemment. Donc, je ne sais pas précisément ce qu'ils sont. On avait entendu parler des AAV donc, qui avaient été développés par Millet et donc qui contenaient dans la capside des fusogènes myomixeurs, myomakers. Et il y en a d'autres avec un tropisme, donc il y a ceux de Millet et puis d'autres qui ont été... donc je ne sais pas s'ils sont sur le marché, si vous pouvez les utiliser. Donc moi, encore une fois, je ne suis pas dans le monde des AAV, donc je ne sais pas comment on se procure ces virus et comment on travaille avec. Et donc pour la relevance, parce que c'est vachement beau ce que vous faites, pour la relevance du système, parce que en fait dans l'organoïde, l'inconvénient, donc ça a des gros avantages. L'inconvénient, c'est que vous ne voyez pas s'il y a des effets délétères après au niveau hépatique, parce que je crois que les effets surtout qui sont importants en clinique au niveau du côté négatif, c'est les effets de l'inflammation au niveau du foie.
Oui, tout à fait.
Donc, comment... en fait, il y a deux questions dans ma question, dans mon commentaire. C'était des autres AAV que vous pourriez vous procurer, et puis comment, dans vos organoïdes, vous pouvez tester aussi le côté négatif, en fait, de cette transduction.
Alors, en fait, oui, il y a plusieurs AAV qui ont été créés pour avoir un tropisme musculaire amélioré, en effet. Donc, certains ont une séquence qui est publiée, qu'on a le droit d'utiliser. D'autres pour lesquels c'est un peu plus compliqué, où il faut demander l'accord et tout ça. Nous, on a le centre de production de vecteurs au sein du laboratoire, donc qui est ici, qui peut nous produire des vecteurs. Donc, si la séquence est publiée, on peut avoir la production de vecteurs qui va derrière assez facilement. Et c'est en cours justement pour avoir accès notamment à 2 sérotypes qui ont été créés récemment pour avoir un tropisme musculaire amélioré. Donc ça, en 2024, on va avoir ces nouveaux vecteurs qu'on va pouvoir tester sur nos modèles in vitro normalement. Et pour la deuxième question concernant du coup les problèmes hépatiques, en effet, c'est quelque chose que j'ai pas forcément précisé, mais le problème avec la thérapie génique et notamment avec les vecteurs AAV, c'est souvent dû à un problème, une toxicité hépatique. C'est quelque chose qu'on pourra pas forcément voir dans nos modèles organoïdes comme ils sont à l'heure actuelle. Du coup, nous, l'idée, c'est vraiment d'utiliser nos modèles organoïdes avant les modèles animaux, en complément des modèles animaux. Donc, en fait, on testerait, on screenerait un nombre d'AAV assez important, on sélectionnerait les plus efficaces qu'on pourrait ensuite aller tester chez les animaux pour voir cet effet toxique chez les animaux pour l'instant. Après, il y a forcément, on pourrait aussi faire des organoïdes de foie pour voir l'efficacité, du coup, sur des cellules humaines en faisant des organoïdes hépatiques à partir d'IPS. Ça existe déjà et c'est déjà fait, donc c'est aussi une idée.
Merci, superbe.
Merci.
Super. Il y a une question encore de Pauline Henrot?
Oui, du coup, merci beaucoup Clément, c'était vraiment super clair et aussi je me doute que c'est énormément de boulot parce que c'est pas de tout repos, notamment le week-end de travailler avec les IPS, voilà pourquoi je n'en fais pas. Non mais du coup ma question, désolée, était, il est vraiment chouette le système commercialisé que je connaissais pas avec les petits piliers et je me demandais si c'est quelque chose que vous alliez faire ou est-ce que c'est une direction que vous pensiez avoir? Est-ce qu'il est possible de mesurer l'intensité de la force contractile dans ce genre de système, justement, de l'organoïde, de faire
Oui.
des mesures comme ça?
Alors oui, c'est possible. Il y a une machine qui est vendue avec ce système de plaques pour mesurer justement les forces de contraction, pour voilà, étudier un peu plus en détail les contractions de cellules musculaires. Et nous, au laboratoire, en parallèle, on crée aussi un autre système pour faire nos organoïdes, qui reprend un peu le même principe avec deux piliers inflexibles, mais où on pourrait ajouter une stimulation mécanique également. Et du coup, on est plus en train de travailler sur ce système-là en parallèle, où il sera possible également d'étudier les contractions, la force de contraction.
Oui, c'est chouette pour relier un peu avec les données fonctionnelles après pour modéliser
Voilà.
la maladie et la guérison. Ok, ouais, c'est
On peut
super.
vraiment avoir un aspect fonctionnel aussi après pour justement, quand on va passer sur le transgène thérapeutique, voir vraiment si
La différence,
y
ouais.
a une restauration phénotype et de...
Et j'ai une petite question de curiosité, mais c'est un peu plus pratique et vous n'êtes pas obligé de répondre. Mais est-ce que vous utilisez déjà des additifs autres que les additifs classiques, sérum, etc., dans les milieux de culture pour promouvoir, enfin voilà, des cues biochimiques, quoi, en plus de déjà les cues mécaniques que vous avez avec les petits piliers?
Alors nous, notre protocole de différenciation, c'est celui qui est publié par Chalpourquier, du coup, qui était dans ma diapo du début sur le protocole de différenciation. Et on n'a pas encore vraiment fait d'optimisation sur ce milieu-là. Mais effectivement, on veut aussi travailler dessus. Ce sera peut-être un projet l'année prochaine pour justement essayer d'améliorer le milieu de différenciation en tant que tel et ajouter d'autres cytokines, d'autres facteurs.
Ok.
Voilà, essayer d'améliorer ça.
Merci.
J'avais vu une autre main levée, ça, c'est bon? Vous avez encore d'autres questions? Très bien. Merci beaucoup à vous deux d'avoir présenté vos magnifiques travaux, du coup.
Merci.
Et,
Merci beaucoup.
et je vous invite à venir pour les prochaines GDR organoïdes. On vous dira, du coup. Merci.
Avec
Merci.
plaisir.
Bonne journée.
Au revoir.
Alors.